Good afternoon, everyone. So, in the last class, we had discussed about catalysis, right? Dia, can you tell me the difference between promoters and poison?
Promoters enhance the reaction and poisons the disease.
Yes, reduces the activity of catalyst, right? Then, which are the two types of catalysis? Anna, children, please switch on your videos.
Yes, ma'am.
Yeah.
Act, so, no, ma'am, what's the question?
Which are the two types of catalysis?
Homogeneous, sorry, homogeneous and heterogeneous.
Yes, homogeneous catalysis and heterogeneous catalysis. Aditya, what's the difference between these two?
Homogeneous both reactants and the
Just
catalyst
1 minute,
are
just 1 minute. Say a little more louder, I can't hear properly.
In homogeneous, both the reactants and catalyst are in same phase.
Okay.
In
Then,
heterogeneous, both are in different phases.
okay. Punya, can you give one example for heterogeneous catalysis?
Haber's process by ion as catalyst.
Yeah, manufacturing ammonia, iron act as a catalyst, right? So here, which one, what about the phase of reactants and catalyst?
Nitrogen and oxygen are in gaseous phase and ion is in solid phase.
Okay. So it's an example for heterogeneous catalysis. Then we had learned the absorption theory of heterogeneous catalysis, right? It consists of 5 steps. Then which are the two important features of solid catalyst, Steffi? Steffi, can you hear me? Yeah. Which are the two important features of solid catalyst? Can you hear me, Steffi?
Yes, ma'am.
Which are the two important features of solid catalyst?
Activity and selectivity.
Yes. So I think we had completed activity, right? Then now you can complete the selectivity. Selectivity. The selectivity of a catalyst is its ability to direct a reaction to yield a product. Okay? Its ability to direct a reaction to yield a product. That means here the reactants are same, hydrogen and carbon monoxide, and using different catalysts we can able to produce different products. In the presence of nickel, methane is formed. Then in the presence of copper with chromium oxide or zinc oxide, methanol is formed. And if copper alone as catalyst, formaldehyde or methanol is formed. Methanol, this one is methanol, this is methane. So from this feature we can write the substance which acts as catalyst in one reaction may fail in another. Okay, that means a single catalyst cannot able to catalyze all reactions. All has its own specific catalyst. That means catalysts are selective in nature. They can able to produce only particular products in particular types of reactions. So these two are the important features of solid catalyst. First one is activity and the second one is selectivity. This is clear, children? Sania, Gayatri, Shivani, Emmanuel, Muhammad Adi, Malavika, Anthony, Naurin, Anna, Michael, all of you switch on your videos. Shall I move to next one, children? Next topic is shape selective catalysis by zeolites. Okay, shape selective catalysis by zeolites. So here it depends upon the shape. The catalytic reaction that depends upon the pore structure of the catalyst and the size of the reactant and product molecules is called shape selective catalysis. So here the pore structure of the catalyst, this I told you in the mechanism, there are certain pores on their surface on which this particles can able to accumulate or concentrate. Okay, so here this catalytic reaction that depends upon the pore structure of the catalyst and the size of the reactant and product molecules, it is called shape selective catalysis. So the size and shape that able to fix in that pore only can accumulate or concentrate on that catalyst surface. Okay, such types of reactions are known as shape selective catalysis. And the zeolites, they are good shape selective catalysis because of their honeycomb structure. Here this white portion, this one is a zeolite. Okay, it's directly obtained from nature and also prepared synthetically. So this one is known as zeolite. It's an example for a shape selective catalyst. Okay, shape selective catalyst. They are microporous. That means two small pores are there. And aluminium silicates with three-dimensional network of silicates in which some silicon atoms are replaced by aluminium atoms, giving aluminium oxygen silicon framework. Children, just wait, there is one mistake there. There are microporous aluminium silicates. Okay, that's fine. They are microporous aluminium silicates with three-dimensional network of silicates in which some silicon atoms are replaced by aluminium. So it is aluminium, then oxygen, silicon framework. So these are the different atoms present in this zeolite. So the zeolite can able to act as a shape selective catalyst. There are certain micropores on their surface and various substances can able to accumulate or concentrate on the surface depending upon their shape and size. If they are fitted to that pore, they can able to accumulate there. Mehak, can you please read the slide about shape selective catalysis?
The catalytic reaction that depends upon the pore structure of the catalyst and the size of the reactant and product molecule is called.
Not audible. Yeah, Mehak?
The catalyst.
Yes, you can read.
Ma'am, is it audible now?
Yes.
Catalytic reaction that depends upon the pore structure of the catalyst and the size of the reactant and product molecule is called shape selective catalysis. Here
Now I can't hear you. Sometimes your voice is low. It's okay, Mehak, it's okay. They are microporous aluminium silicates with three-dimensional network of silicates in which some silicon atoms are replaced by aluminium atoms. That's it. Okay. Have you completed? So you have to note down these points. It depends upon the pore structure of the catalyst and the size and shape of reactant and product. Okay. The pore structure of catalyst and the size and shape of this reactant and product. That's why it is known as shape selective catalysis. Shall I move to next slide? Have we completed? Yes. No? Okay. Emmanuel, then you can tell me whenever you had complete. Finished? Yes, okay. Okay. Then no need to write again. This is the same point that we write in the beginning. Okay. Reactions taking place in zeolites depend on the size and shape of reactant and product molecules as well as upon the pores and cavities of zeolites. Now I told you it's already found in nature and also we can synthesize this material, zeolite. Okay. ZSM5. It's a type of zeolite catalyst that is used in petroleum industry to convert this alcohol directly into gasoline, petrol, by dehydrating them to give a mixture of hydrocarbons. So this ZSM5, it's a type of zeolite catalyst used in petroleum industry. It is able to convert this alcohol directly into gasoline or petrol by dehydrating them to give a mixture of hydrocarbons. What do you mean by dehydration?
Loss of water.
Loss of water, right? So in this alcohol it consists of OH group. Okay, ROH. R stands for any chain. Okay, any hydrocarbon chain, CH3 or CH3, CH2, any long chain like that. So here it is dehydration, that means lose water molecules and it will become R itself, right? So here the zeolite catalyst ZSM5, it is used in petroleum industry, convert alcohol directly into gasoline petrol by dehydrating them to give a mixture of hydrocarbons. R stands for hydrocarbons. So here a mixture of hydrocarbons is obtained after the reaction. So here this zeolite ZSM5 acts as a catalyst. That means provide a surface for the chemical reaction to take place and makes the reaction faster. And this powdered form of zeolite is used for different reactions. Okay, we can able to convert it into powdered form. What's the reason? We have learned one factor behind that. Do you remember? Why powdered form is preferred? Rijas, can you tell me? Why powdered form are preferred for these reactions?
Surface area will increase.
Very good. Surface area increases, right? That's why. Okay. Nevin, can you please the second and third point?
Found in nature as well as synthesized.
Yeah.
Terrorism 5 zeolite catalyst is used in petroleum industry. Convert alcohol directly into gasoline by dehydrating them to give a mixture of hydrocarbons.
Okay. Finish, children? All of you? Gauri?
1 minute, ma'am.
Okay, okay.
Yes, ma'am.
Yes. All of you switch on your videos. Evelyn, Naurin, Srihari, Anthony, Malavika, Shivani, Muhammad Adim, Aryan, Sania. Okay, next we are moving to next topic, that is enzyme catalysis. Okay, enzyme catalysis. What do you mean by enzyme? Vidur? What do you know about enzymes? Yeah, unmute and tell me.
Chemical substances
Yeah, substances produced by who can able to produce enzymes? Human beings? Plants? Animals?
human beings
Yeah, plants and animals. Along with that, human beings also able to produce certain compounds. It is known as enzymes. Okay. So this enzyme can also act as catalyst. So such reactions are known as enzyme catalysis. Enzymes, they are complex nitrogenous organic compounds which are produced by living organisms, plants, animals, including human beings. Okay. Complex nitrogenous organic compounds. Nitrogenous organic compounds which are produced by living organisms. And they are certain protein molecules of high molecular mass and form colloidal solutions in water. Protein molecules of high molecular mass. They are protein molecules of high molecular mass and form colloidal solutions in water. What do you mean by colloids? Colloids. Can you tell me one example for such a colloid? Anyone? Yeah. Michael, can you tell me? Michael, can you tell me one example for a colloid? No.
What's the question?
Can you give me one example for colloid?
Colloids are solutions which have some impurities in them.
Yeah, can you give one example? No. Okay, it's okay. Milk is considered as an example for a colloid. Okay, ink. So colloid is a heterogeneous mixture in which it can able to scatter the beam of light. Then the particles are too small but they are big enough to scatter light. That's it. We cannot see the particles with naked eyes. Okay. But they can able to scatter the beam of light. Do you remember the Tyndall effect that you have learned in 9th, right? So this colloidal particles can able to scatter this beam of light and this path of light is visible. That is known as Tyndall effect. Okay, so colloids are certain types of mixture. It's a heterogeneous mixture. One example, milk. In milk there are different types of substances, right? Protein, fat, water, etc. Isn't it? They are not in uniform composition. They are non-uniform. That's why they belongs to heterogeneous mixture. Okay. So here these enzymes, they also able to form colloidal solutions in water. Do not completely mixed with water, they are certain mixture with water in the form of colloids. So protein molecules of high molecular mass and form colloidal solutions in water. That's about enzymes. Then it's an effective catalyst connected with natural processes. Okay. Enzyme, it can act as an effective catalyst in almost natural processes. The reactions that occur in the bodies of animals and plants to maintain the life processes are catalyzed by this enzyme. So these processes are known as biochemical catalysis. Different biochemical actions are taking place in our body, right? Digestion, respiration, then circulation of blood. So many things are there. So this enzymes, the major role of this enzyme to catalyze all these reactions to maintain the life process. Okay, the reactions that occur in the bodies of animals and plants to maintain the life process are catalyzed by enzymes. It is known as biochemical catalysis. It is known as biochemical catalysis. And these enzymes are termed as biochemical catalyst. Biochemical catalyst. Have you learned about pepsin, children? Pepsin in stomach, biology students? Yes, right? Yeah. Pepsin is an enzyme that present in stomach. It can able to convert proteins into amino acids. Okay. Similarly, so many enzymes are there in the body of plants and animals. The major role of all enzymes to catalyze the biochemical, that means the reactions that able to maintain the life process. That's it. This is clear? Sharon, can you please read these points?
Yes, ma'am.
Yeah.
Enzymes, complex nitrogenous organic compounds which are produced by living organisms, plants and animals.
Okay.
Protein molecules of high molecular mass and form colloidal solutions in water. Effective catalysts connected with natural processes. Reactions that occur in the bodies of animals and plants to maintain the life process are catalyzed by enzymes is known as biochemical catalysis.
Yes.
Enzymes are termed as biochemical catalysts.
Okay. Have you completed? All of you? Yes. Then the first enzyme, okay, first enzyme, it was synthesized in laboratory in 1969. Not only in the bodies of plants and animals, we can able to synthesize certain enzymes. Okay. And the first production of certain enzyme, it's taken place in laboratory in 1969. And we can also obtain this enzyme from living cells in the form of certain crystals. We have learned that these enzymes can be obtained from living organisms, right, including plants and animals, and it's in the form of a crystal. Clear? It's in the form of a crystal. Then these are certain examples of enzyme catalyzed reactions or biochemical catalysis. First one is inversion of cane sugar. Okay, inversion of cane sugar. That means sucrose. Cane sugar, otherwise known as sucrose. Here this invertase, that's the name of the enzyme. Invertase enzyme converts this cane sugar into glucose and fructose. Invertase enzyme converts cane sugar into glucose and fructose. Both have the same chemical formula. Glucose and fructose. This one is sucrose, C12H22O11, or otherwise known as cane sugar. Cane sugar. And the second example, conversion of glucose into ethyl alcohol. Conversion of glucose into ethyl alcohol. Here the zymase enzyme, the name of enzyme is zymase. Zymase enzyme converts glucose into ethyl alcohol and carbon dioxide. Ethyl alcohol and carbon dioxide. Conversion of glucose into ethyl alcohol and carbon dioxide by the enzyme zymase. Finished?
No.
Okay. Evelyn, can you please read the examples?
Yes, ma'am.
Okay.
Inversion of cane sugar.
Yeah.
Invertase enzyme convert cane sugar into glucose and fructose. Sucrose plus water will be giving glucose plus fructose.
Yes.
And conversion of glucose into ethyl alcohol. Zymase enzyme convert glucose into ethyl alcohol and carbon dioxide.
Yes.
And glucose will be getting converted into ethyl alcohol plus carbon dioxide under the
The
enzyme
presence of enzyme zymase. Finished, right? Then another examples for the same enzyme catalysis. I told you about pepsin enzyme, right? In stomach, this pepsin enzyme converts proteins into amino acids. This peptides means that is amino acids. Okay. While in intestine, the pancreatic trypsin, it's the name of another enzyme, pancreatic trypsin, that convert proteins into amino acids by hydrolysis. Peptides, it's a form of amino acid, but the most shortest form, that's I think amino acids. Isn't it, children? What about biology students? Amino acids is the simplest form of this protein. So amino acids join together to form this peptides, then polypeptides, then proteins. That's it. So in stomach, this pepsin enzyme converts proteins into peptides, while in intestine, the pancreatic trypsin converts proteins into amino acids by hydrolysis. So here two enzymes are there. First one is pepsin enzyme and the second one is pancreatic trypsin. Pancreatic trypsin. And even in the conversion of milk into curd, there is one enzymatic activity. That's enzymatic reaction brought about by lactobacilli enzyme. It was produced by lactobacillus bacteria. You know that, right? So here this bacteria can able to produce lactobacilli enzyme that helps for the conversion of milk into curd. Lactobacilli enzyme. There are more examples in your textbook. Okay, I just included only 4 examples. But here starch can be converted to maltose by diastase enzyme. Then maltose can be converted to glucose by maltase enzyme. It's in page number 133. Page number 133. While learning, you have to just include these examples also, right? 3, 4, 5. Examples 3, 4, 5 not included in this slide. And just turn the page. In page 134 also some examples of enzymatic reactions. Enzyme invertase, source is yeast, sucrose is converted to glucose and fructose. Next one is zymase, table 5.2. Okay, that's also important. You can try to learn. Different enzymes are there: invertase, zymase, diastase, maltase, urease, pepsin, etc. And the sources in which, that means sources that can able to produce this enzyme, and the enzymatic reaction, which one is converted to which one. Okay. This is clear? Have we completed? Okay, then we are moving to the characteristics of enzyme catalysis. There are different features or characteristics of this enzyme catalyzed reactions or enzyme catalysis. First one, most highly efficient. These reactions are most highly efficient. That means even one molecule of an enzyme may transform 1 million molecules of reactants per minute. A small amount of catalyst is required, especially in the case of enzymes. Even one molecule of enzyme can able to change millions of reactants to products. Okay, so it is most highly efficient. Most highly efficient. And the second one, it is highly specific in nature. Each enzyme is specific for a given reaction. We have learned the names of specific enzymes, right? For each reaction, there are certain specific enzymes. Only that enzymes can increase or interfere in that reaction. One catalyst cannot catalyze more than one reaction. One catalyst cannot catalyze more than one reaction. So this is the first point, most highly efficient, and the second one, highly specific in nature. Highly specific in nature. This one most highly efficient, and the second one is highly specific nature. Lydia, can you please tell me the first two characteristics of this enzyme catalysis? Please switch on your video.
Most highly efficient.
Yes.
One molecule of an enzyme may transform 1 million molecules of the reactant per minute. And second one, highly specific nature.
Yeah.
Enzyme is specific for an given reaction. That is, one catalyst cannot catalyze more than one reaction.
Okay. Shall I move to third point? Gauri, finished?
Yes, ma'am.
Okay. Then third one, highly active under optimum temperature. Highly active under optimum temperature. The rate of an enzyme reaction becomes maximum at definite temperature. It is known as optimum temperature. Okay. The enzymatic activity that is maximum at a particular temperature, that one is known as optimum temperature. And the optimum temperature range for enzymatic activity, it is 298 to 310 Kelvin. Okay, 298 to 310 Kelvin. And in our human body, the enzyme catalyzed reaction, that is near to 310 Kelvin. Highly active under optimum temperature. The rate of reaction, that means rate of enzyme reaction, becomes maximum at a particular temperature. It is known as optimum temperature, and it has a range of 298 Kelvin to 310 Kelvin. And in our human body, all this enzyme catalyzed reactions take place at 310 Kelvin. 310 Kelvin temperature, it is suitable for all reactions. So can you tell me it in degree Celsius? It's in Kelvin. How can we convert this into degree Celsius? Tell me. Achyut, can you tell me how can we convert Kelvin to degree Celsius?
Like minus 273.
Yeah, that's it. 310 minus 273. So it is 37 degree Celsius. Okay, 37 degree Celsius. And the second point, it is highly active under optimum pH. It is highly active under optimum pH. What do you mean by pH? pH. The nature of substance, right? It can be measured using a scale. It is known as pH scale. And what about the value of acidic substances? In this pH scale, is it greater than 7 or less than 7?
Less than 7.
Yeah, less than 7. Then what about alkaline substance? Greater than 7. And for neutral, it is 7. Right? So here, the rate of an enzyme catalyzed reaction, that is maximum at a particular pH, it is called optimum pH, and which is between the pH values 5 to 7. 5 to 7. That means acidic in nature. Slightly acidic. Okay. So it is highly active under optimum temperature and optimum pH. Vidur, can you please read the slide?
Highly active under optimum temperature.
Yeah.
The rate of an enzyme reaction becomes maximum at a definite temperature called the optimum temperature. The optimum temperature range from enzymatic activity is 298 to 310 Kelvin.
Okay.
Highly active under optimum pH. The rate of an enzyme catalyzed reaction is maximum at particular pH called optimum pH, which is between pH values 5 to 7.
Okay. Shall I move to next one? Yes. The next point, increasing activity in the presence of activators and coenzymes. We have learned about promoters, right? That can able to enhance the activity of a catalyst. Here there are certain substances, they are known as activators and coenzymes. They can able to increase the activity of enzyme. Okay? Activators and coenzymes. Enzymic activity or enzymatic activity, it is increased in the presence of certain substances known as coenzymes. Coenzyme. So it's the non-protein part in the enzyme. Okay? Coenzyme. It is the non-protein part in the enzyme. It can able to increase the activity of this enzyme. And the second one is known as activators. They are usually metal ions. Okay, metal ions, they are weakly bonded to enzyme molecules, increase their catalytic activity. Example, amylase is an example for an enzyme. It can increase their activity in the presence of Na plus, sodium plus ion. So, two types of substances that can able to increase the activity. They are known as activators and coenzymes. Activators and coenzymes. Coenzymes, they are the non-protein part of this enzyme and able to increase the enzymatic activity. Then activators are certain metal ions weakly bonded to this enzyme. Same function here, increase the enzymatic activity. So this Na plus ion is an example for this activator in amylase enzyme. Amylase enzyme. Then we have learned about poisons, right? Substances that able to decrease the rate of catalyst. So here this inhibitors and poisons also same function. They can able to reduce the activity of enzymes. Enzymes are also inhibited or poisoned by the presence of certain substances. Inhibitors or poisons interact with active functional groups on the enzyme surface and often completely destroy the catalytic activity of the enzyme. I told you enzymes have certain micropores, right? This is considered as a pore, and if this inhibitor or poison will occupy this position, then no other molecule or substrate can accumulate or concentrate on the surface. Okay. So this inhibitor and poison will attach to the pores of that catalyst or enzyme. Then it will destroy the catalytic activity of the enzymes. So these are the different characteristics of enzyme catalysis. They are most highly efficient, then highly specific in nature, highly active under optimum temperature and optimum pH. Then this activators and coenzymes can able to increase the enzymatic activity. Then inhibitors and poisons can able to decrease the enzymatic activity. As an example for this one, you can write use of certain drugs. They will decrease the production of enzymes. That means they will occupy the pores of this enzyme and reduces the enzymatic activity. Have you completed, children? Yes, right?
No, ma'am.
Okay, now it's time. I will share the screen once again in the next class. Okay. Yes. So can you tell me, please switch on your videos, children. Meenakshi. No. Then what about Muhammad Adi? Present, right? Nevin? Nia? Shivani?
Yes.
Okay. Then thank you, children. All of you read this portion very well. Okay. Okay. Then thank you all. See you in the next class. Bye. Thank you.